So in this topic, we're going to try and answer the questions of what is meant by the term genome, how PCR can be used to amplify DNA samples, and how then these samples can be used to predict the amino acid sequence of proteins using gene sequencing, and also in forensic science to identify criminals and to test paternity using DNA profiling. In the last 50 years, one of the fastest growing areas of science has been our understanding of genetics and the genome. The genome is the total of all the genetic information in an organism. In eukaryotic cells, this includes the DNA in the mitochondria and/or chloroplast, as well as the nucleus. In terms of the human genome, we now know that it is over 3 billion base pairs long. However, only about 2% of that DNA actually codes for proteins. There are over 20,000 genes, of which we have worked out the function of about 15,000. We are very interested in the genome of organisms as it tells us not only how it functions, but its evolutionary history and classification as well. In this topic, we're going to look at gene sequencing, and also we're going to do something called DNA profiling. A DNA sample may be very, very small to start with. So in order to sequence it or to find a profile, the first step will be to amplify that sample using something called the polymerase chain reaction, or PCR. Now, as many applications in gene technology, for example, cloning of gene fragments to analyze genetic diseases, identification of contaminant foreign DNA in a sample, or amplification of the DNA for sequencing or profiling, as we've mentioned. What it does is it takes a small sample of DNA and rapidly produces large quantities of it. The process involves 3 steps, and you're going to need a thermal cycler, some primers, free DNA nucleotides, buffer, and DNA polymerase. Step 1 involves separating the DNA strands via heating to 95 degrees C in the thermal cycler. This denatures the hydrogen bonds and then exposes the bases. Step 2 is something called annealing. The mixture cools to 55 degrees C. Primers are added to the start of the target genes. And then step 3, elongation, is you heat it back up again to 72 degrees C, and DNA polymerase replicates the DNA using complementary base pairing. The cycle then repeats over and over about 30 times until there are about 1 billion copies, which takes about 3 hours. The DNA polymerase used is a special type of DNA polymerase called Taq polymerase. It comes from a bacterium called Thermus aquaticus, which lives in hot springs and hydrothermal vents, so has evolved a DNA polymerase enzyme that can withstand high temperatures. It is very thermostable. Now, the benefit of this is that you can use it in PCR where you're heating up to those high temperatures and it won't denature the enzyme. Once you've done PCR, you can then go about further processes such as DNA sequencing. DNA sequencing is the process of working out the order of bases in a strand of DNA.